now repeat. Gone this way. Okay. Dear students, today we are going to the discussing about, so in last topic in our intermediate textbook and 11th and 12th chapter, so that is the biotechnology. So in our intermediate exam, one of the essay question, 4 marks question and 2 marks questions are appear. In this lesson having the 11th and 12th chapter in our IP textbook. 11th chapter is biotechnology process, principles and process. And second lesson, biotechnology applications. So what are the applications of the biotechnology? So దీనిలో మనకి ఒక 8 marks and 4 marks, 8 marks and 2 marks, 10 marks. From this chapter, 4 marks question and 2 marks, total 6 marks. So total 16 marks for your intermediate examination. And also, especially in 2020 NEET, 5 bits are appear, 5 plus 1, 5 bits from biotechnology principles and process, and another bit from to the biotechnology applications. Totally 5 bits are appear in that final examination, plus 1, 6, total 6 into 4, 24. So 24 marks awarding to this biotechnology, most important for NEET. And I'm saying 2 plus 2, 4 marks kachitanga, 4 bits compulsory appear within the MCET examination. So that's why today we are discussing about the biotechnology principles and process. Before going to that biotechnology principles and process, what is biotechnology? So that biotechnology is nothing but applications of the microbiology. But what is microbiology? The study of microorganisms. The study of microorganisms is called microbiology. So in that microbiology, what are the organisms are present in microbiology? Mainly bacteria, virus, algae, fungi, and protozoans. So all these ones are studied under the microbiology. But what are the events those are studied in this biotechnology? How those microbes are useful to human beings. Microbes, their cells, their cell constituents, their properties, cell properties, or everything is useful to the production of the products. In large scale production, not only small amount, in industrially large scale production. So to our daily needs, or maybe food products, or maybe medicines, or everything are producing by using the microbes. That study is simply said to be biotechnology. Right, in this one, large number of techniques are also used to production of the new organisms. Our new organisms produce చేయడం కోసం different techniques వాడుతూ ఉంటాం. So when we are observing in that biotechnology, in that 11th chapter, so that biotechnology principles and process. So biotechnology principles and process. So those are discussing within the biotechnology principles and process. In that biotechnology principle and process, so one of the specialized technique is present, that is called genetic engineering. So simply said to be GE, genetic engineering. What is the meaning of genetic engineering? So genetic engineering means, so we are producing into new plants by using some of the special techniques. So what happened during the genetic engineering process? So we are isolated the desirable genes from to the one plant. So these are the desirable genes. So we are isolate the desirable genes from one plant. Those are introducing into another plant. Those are introducing into the another plant through the vectors. Through the vectors. So we are actually in genetic engineering process. We are isolated the desirable genes. Those desirable genes are isolating from to the one plant. Those are introducing into another plant through the vectors. So before going to that genetic engineering process, or you can see the what is the gene. So you know about the knowledge about that gene. So what is the gene? So the gene is a hereditary unit. That gene is a hereditary unit. But physically, how can you available that gene? So that gene is nothing but part of chromosome. The gene is nothing but part of chromosome. Or gene is nothing but part of DNA. So the DNA containing sequence of the nucleotides. What are those 4 types of nucleotides? A, T, G, C. Those are sequentially arranged. So the DNA is the double stranded. In this one, those 2 strands, polynucleotide strands containing the different types of nucleotides. The sequence of the nucleotides are also considered as the sequence of the nucleotides is also considered as a gene. Nothing but sequence of the nitrogen bases, adenine, guanine, thymine, cytosine. The type of sequence of the nitrogen bases. So those are also considered as a gene. Such type of genes are isolating from to the one plant. Those are introducing into another plants through the another DNA. The DNA is called vector DNA. So those are introducing into another plant. A new plant is produced. That newly producing plants are called transgenic plants. Those plants are called transgenic plants. And also those plants are simply said to be GM plants. Genetically modified plants. Those organisms are also called GMOs, genetically modified organisms. So such type of organisms are producing through the biotechnology process are nothing but genetic engineering process. So this type of genetic engineering process, it is one of the laboratory technique. In this one, a new that desirable genes are combined with vector DNA to form into one of the new DNA. That new DNA is called rDNA. rDNA means recombinant DNA. So recombinant DNA. That's why genetic engineering is also called rDNA technology. Genetic engineering is also called, what is that name? rDNA technology. So now itself, you can be see in this lesson. So how that first rDNA was constructed by Coher and Stanley. So Coher and Stanley, so they were constructed. So Stanley Cohen and Herbert Boyer. Stanley Cohen and Herbert Boyer, they were first constructed that rDNA. So Stanley first rDNA was constructed by Stanley Cohen and Herbert Boyer. Herbert Boyer. They were first constructed that rDNA, recombinant DNA, a novel DNA, combination of the 2 types of genes. So they were isolated antibiotic resistant genes. They were isolated antibiotic resistant genes. So those antibiotic resistant genes are isolating from to the organism by using specialized enzymes. The enzymes are called restriction endonuclease enzymes. So the restriction Endonuclease. That restriction endonuclease enzymes was mainly helpful to the DNA fragment or resistant genes containing antibiotic resistant genes containing the DNA can be cut into small fragments. Cut the small fragments with the help of okay. So those restriction endonuclease enzymes are mainly identify the particular DNA sequence and cut the DNA into small fragments. Hence, these restriction endonuclease enzymes are called molecular scissors. So that molecular scissors are also important to mass question. So those are the restriction endonuclease enzymes. Those restriction endonuclease enzymes are identify the particular sequence on the DNA and cut the DNA into small fragments. That antibiotic resistant genes are introducing into the plasmid of the one bacterium. That bacterium is called Salmonella typhimurium. Salmonella typhimurium. So in that Salmonella typhimurium, they were isolated that plasmid, a circular double-stranded naked DNA present within the bacterial cells in addition to the gene of work. That is called plasmid. That plasmid is isolated from to the Salmonella typhi. So that plasmid is cut into small fragments with the help of restriction endonuclease enzyme or molecular scissors. Afterwards, that antibiotic resistant genes are introducing into the plasmid of the Salmonella typhi. So Salmonella typhi plasmid loki, so our desirable antibiotic resistant genes are introduced, but those are linking together with the help of another enzyme. That enzyme is called DNA ligase. A newly constructed DNA is formed. That DNA is called rDNA. So recombinant DNA. The type of recombinant DNA is introducing into that was carried out within the laboratory, that is not able to replicate when that is present within the laboratory. So that is introducing into another bacterium cell. That bacterium cell is E. coli, Escherichia coli. So why that rDNA is introducing into the Escherichia coli? So the Escherichia coli and Salmonella properties are more or less similar. That's why. So that newly constructed first rDNA antibiotic resistant genes containing recombinant DNA is introducing into another bacterium. That bacterium is called E. coli. So this is the rDNA. That rDNA is the combination of the genes of antibiotic resistant genes and also genes of that Salmonella. Genes of the Salmonella and producing into new DNA. That new DNA is called recombinant DNA, that is introducing into the E. coli cells. In that E. coli cells, those are undergoes to replication, will producing into multiple copies of our desirable genes. That process is called gene cloning. That multiple copies of the genes are produced. That process is called gene cloning. The gene cloning process is carried out within the E. coli. So this is the construction of the first rDNA by Stanley Cohen and Edward Boyer. That antibiotic resistant genes were first introducing into the plasmid of the which one? Salmonella. Afterwards, those circular DNA is modified into linear DNA and cut the DNA into small fragments with the help of special enzymes. Those are called restriction endonuclease enzymes. Those are identify the particular sequence and cut the DNA into small fragments. Hence, these are called molecular scissors. Afterwards, the antibiotic resistant genes are introduced into the plasmid DNA. So, by linking together with the help of another enzyme, that enzyme is called DNA ligase. Those are helpful to the attachment of that antibiotic resistant genes with the plasmid vector DNA. So a new constructed DNA is formed, that is called rDNA, are also said to be recombinant DNA. Okay, so this is the first one. Next, for this rDNA technology process or genetic engineering process, some of the operators, some of the tools are required for to the process. So this is the most important essay question for your IPG. Tools for rDNA technology. So what are the tools? Those are mainly useful to the process of the rDNA technology. So first one, restriction enzymes. Second one, DNA polymerase. Third one, DNA ligase. And fourth one, main important, vector DNA. And fifth one, so host cell, competent host cell. So these are the main important tools which are required for rDNA technology. So these tools are mainly important for rDNA technology. So first one, restriction enzymes. So in 1969, there were two specialized enzymes are recognized in biotechnology. One enzyme is by Nathans and co-workers. So one enzyme can inhibit the growth of the viruses within the bacterial cell. So another enzyme is helpful to the multiplication process of the linking of the thymine to DNA. So ikkada manaki first enzyme is important. But first enzymes are, so those enzymes are mainly naturally occurs within the bacteria to prevent the growth of bacteriophage, to prevent the attach of that bacteriophage. You know that one bacteriophages, those are the viruses which attack on the bacteria. Those are called bacteriophages. వాటిని ఆపడం కోసము బ్యాక్టీరియా సెల్లో ఒక ఇన్హిబిటరీ ఎంజైమ్ ఇస్ డెవలప్డ్. దట్ ఎంజైమ్స్ ఆర్ కాల్డ్ restriction enzymes. So those restriction enzymes are, so the first those restriction enzymes are mainly isolated from the prokaryotic cell, especially in bacteria. So in that bacteria, more than 900 restriction enzymes are isolated. Restriction enzymes. Those restriction enzymes are isolated from to the bacterial cells. Those restriction enzymes mainly identify the particular sequence of the DNA, and the DNA cut into small fragments. So that restriction enzymes are identify the particular sequence on DNA. Those sequences are called recognition sequences, or those are called restriction sites. Those are called restriction sites, and that sequence is called restriction sequence. Those sequences are also said to be palindromic sequence. Those sequences are also palindromic sequence and cut the DNA into small fragments. That enzymes are called restriction enzymes. So the type of restriction enzymes are belongs to class nuclease. Those are belongs to the class nuclease. So in this one, two types of restriction enzymes are present. Those are the belongs to the class nucleases. So in that restriction enzymes, those are identified the particular sequence of the DNA. That sequence is called palindromic sequence. Those sites So restriction sites and cut the DNA on the restriction sites. So those are belongs to class nucleases. In that nucleases, two groups are there. First one is restriction exonucleases. Exonucleases, and second one is the restriction endonucleases. Restriction endonucleases. What is the difference between exonucleases and endonucleases? When you are observing that DNA fragment, on the DNA fragment, you know that one 5' 3' or 3' 5' between that complementary hydrogen bonds, you know very well, ATGC ఇలా ఉంటాయి కదా. సో దీనిలో చూస్తే దట్ రిస్ట్రిక్షన్ ఎక్సోన్యూక్లియస్ ఎంజైమ్స్ కెన్ ఐడెంటిఫై అండ్ కట్ ద DNA into terminal positions, that cut the DNA on that terminal position, hence those are called restriction exonucleases enzymes. And another one, restriction endonuclease enzymes. So that should be there in the name. So those are identify the particular sequence within the DNA. Those particular sequence within the DNA. So those sequences are called palindromic sequence. So what is the meaning of palindrome? So 2020 NEET bit. So identify the palindromic sequence. So important NEET bit. So in that palindrome, so palindrome is a sequence of the nucleotides. So six are many palindromic, 6 are many nucleotide base pairs. So those are same in the nuclear both strands, but read as opposite direction. So palindrome is a sequence of the nucleotides. Those are similar in both strands, but read in opposite direction. For example, Malayalam undanukondi. So you can read as same. So this side it is a Malayalam. So this side also Malayalam. Okay na? Chudandi. Okay. So in both directions it is read as same. The type of sequence is called palindromic sequence. What is palindrome? A sequence of the nucleotides which are similar in both strands, but those are read as opposite direction. For example, one of the restriction endonuclease enzyme is there. That enzyme is called restriction endonuclease enzyme. Important to mass question also, for important to the NEET. ECO R1 లేదా RI. So that is the one of the restriction endonuclease enzyme. Restriction endonuclease enzyme. That enzyme can identify the palindromic sequence. ఎవరు అంటే GAA-TTC. So that is also identify the GAA-TTC. So you know that one ATGC, A double bond T, G triple bond C, G C T T A Z 5' to 3', 5' to 3'. Okay, you can see this one GAA-TTC. In this side also GAA-TTC. So this type of sequence is called palindromic sequence. So that ECOR1, E stands for Escherichia, that is the genus name. CO stands for coli, that means species name. And also R means R1, that is the sequence of the restriction enzyme within the E. coli. So actually full name is CORY13. That is the true name of that ECOR1. So E stands for genus name, Escherichia. CO stands for coli, Escherichia coli. So R stands for restriction enzyme, That is the serial number of the restriction enzyme within the E. coli. So the type of ECOR1 is a one type of restriction endonuclease enzyme that can identify the palindromic sequence GAA TTC and cut the DNA in between guanine and adenine. So that can be identify the palindromic sequence and identify the restriction site and cut the DNA in between G and A. So between the guanine and adenine and form into small fragments of the DNA. The type of, so they can be cut into the guanine and adenine phosphoester bonds. They cut the phosphoester bonds. The type of restriction of endonuclease enzymes which are identified, those endonuclease enzymes are identified particular palindromic sequence and cut the DNA into small fragments. That's why those are called molecular scissors. Those are called molecular scissors. So those molecular scissors are useful to cut the DNA into small fragments. They were cut that fragment, those are cut the DNA into, so two types of cuttings. One cutting is called even cut and another cut is called staggered cuts. So that restriction endonuclease enzymes can identify the palindromic sequence. So palindromic sequence and cut the DNA into fragments. Those are, they can be carried out two types of cuttings. One is called even cut and another one is the staggered cut. What is the meaning of even cut? The two strands of the DNA can cut into the same location. The two strands of the DNA can cut in on the same location. The type of cuts are called even cuts, will producing into those DNA fragments. So those DNA fragments does not able to formation of the complementary hydrogen bonds to the another DNA fragments. The type of ends are called blunt ends. Those ends are called blunt ends. Those are not able to form the complementary hydrogen bonds to the another DNA. But another type of cuttings are there. Those are called staggered cuts. What is the meaning of staggered cuts? The DNA, that restriction endonuclease enzymes can identify the palindromic sequence and cut the DNA into different locations. And cut the DNA into different locations will form into this type of fragments. Those cuttings are called staggered cuttings. Those ends are called, this type of fragments are called sticky ends. Sticky ends. So those sticky ends are formed. So those are easily form the complementary hydrogen bonds. Those are easily produce complementary hydrogen bonds to the between the two DNA's cutting strands. The type of cuttings are called staggered cuts. The type of ends are called sticky ends. Those are easily attached to the another DNA fragment. Okay, so these are the restriction endonuclease enzymes. Okay, so next come to that another enzyme, DNA polymerase. So before going to the DNA polymerase, first of all you can see the DNA ligase. So when we are isolated that vector DNA from to the one bacterium, that can be cut into that circular DNA can cut into the, so open it with the help of restriction endonuclease enzyme, our rDNA is introduced. So our rDNA or desirable gene. So the desirable gene is introduced into the vector DNA with the help of enzyme DNA ligase. So those, so that cutting desirable genes are linking to the vector DNA with the help of an enzyme. What is that enzyme name? DNA ligase. That DNA ligase is also useful to the attachment of that DNA fragments or desirable genes. Those are obtained through the which type of cuts? Staggered cuts. Those ends are called sticky ends. Those are attached to the vector DNA with the help of enzyme restriction endonuclease. With the help of enzyme, with the help of enzyme DNA ligase. That enzyme is the DNA ligase. A new DNA is produced. That new DNA is called rDNA, recombinant DNA. So when that recombinant DNA is present within the laboratory, that is not able to replicate, does not produce the multiple copies of our desirable genes. Kani, you required many multiple copies of the desirable gene. To production of the multiple copies of the desirable gene, for gene cloning process, we are using another DNA. That DNA is called vector DNA. Din tarvata idi chebudama. So the DNA is called, what is the DNA? Vector DNA. Okay, so you can go to that vector DNA.